Hey everyone, it's time to learn our next skill in solving genetics problems. So, to do our next type of problem, we're going to have to learn how to do this. We learned it when we were a sophomore, so it shouldn't be too bad. That is finding allele combinations. So I've wrote some alleles down here, some genotypes. The first three are for monohybrid crosses where we're just looking at one trait at a time. So in this case, we're looking at gene A, which comes in two possible alleles, big A and little a. And then in the second line, we're looking at two traits at once. So that's for dihybrid crosses. So gene A and gene B, both of them come in two different alleles, the capital letter and the lowercase letter. So the law of independent assortment says that you can't give both of your alleles to your kids or to your gametes, so they must be separated during meiosis. So what are the possible combinations that they would end up in your gametes? So let's start with the first one, big A, big A. What are the combinations? Well, nothing too surprising here. It's just big A. So what I do is I write that and then I circle it to signify that that's going into a gamete. Over here we have the heterozygous condition. So again, those must be separated. You can't give both the big A and the little a to your kids. You must separate them into different gametes. So the gamete possibilities are big A and little a. And then again, I circle them to show that they're going into separate gametes. Over here, little a, well, the only possibility that you'll find in your gametes is the little a. So then I circle that. So those are pretty easy. We've already been using that skill when we do monohybrid crosses. How about when we do dihybrid cross? Because that's up next, following two traits at one time. One of the first steps in doing dihybrid crosses is finding the allele combinations so that we can put those into Punnett squares and do that successfully. So it's called the FOIL method. You've used it in math before. So FOIL stands for first, outside, inside, last. And in math, you may write them more than once. In genetics, I just write them once. So for this first one, if I use the FOIL method, first, big A and big B, then anything else I say after that, because for both traits, they're homozygous, it's always going to be big A, big B, big A, big B, big A, big B, and big A, big B if you were watching where I was pointing there. So all the gametes are going to have a big A and a big B in them. So whatever the traits are that those genes symbolize, there'll be one of each in every gamete that you make. It gets a little more interesting when you finally have traits that are heterozygous. Like here, we'll use big A, big B as our first combination. Then, well, big A, big B, that's a repeat. I'm not going to write that twice. So go to the next one. Inside, little a, little a, big B. It's kind of hard to write here on the Jamboard, but I circle them to symbolize those are going into gametes. So there's only two possible combinations for this one, big A, big B, and little a, big B. Now for this next one, it's heterozygous for both traits. So first is big A, big B. Outside, big A, little b. Inside, little a, big B. And last, little a, little b. So there are four possible gamete combinations for this one when they're heterozygous for both traits. Get a little more variety in your gametes that way when it's heterozygous. So that's how you do that, and that's actually how you will start many dihybrid crosses. Sometimes you'll maybe only have one possibility, sometimes you'll have all four. If we did a trihybrid cross, such as if I did big A, big B, little b, and then how about big C, little c? For that one, if you do the FOIL method, you'll come up with 8 possibilities for that one, but luckily we won't do those yet. We'll stick with monohybrid and dihybrid crosses for the moment. Let me know if you have any questions on how to do that part. Then we'll move back to our notes. So we'll be finishing up our Mendel notes. We've talked about monohybrid crosses. Now we'll talk about the dihybrid crosses, which is another one of Mendel's research areas, following 2 different characteristics or traits at a time, such as with pea plants, pea seeds. He could have used 2 traits like green and yellow seeds, like we see in the picture, and then round and wrinkled seeds. In this, he's going to work out some more of these genetics rules that we need to follow. All right, let's follow this. We have a true breeding or homozygous yellow round pea, and we cross it with a true breeding green wrinkled pea. Again, true breeding means homozygous. So both of these peas are homozygous for both of their traits, yellow round, green and wrinkled. So he crosses them, and 100% of his F1 generation is yellow and round. So that must mean yellow is dominant and so is round, right? So we'll take it a step further and see what happens in the F2 generation. We allow those to self-pollinate. In the F2 generation, we get our mix back. 9 out of 16 are yellow and round. 3 out of 16 green and round. 3 out of 16 yellow wrinkled, and 1 out of 16 green and wrinkled. There's our F2 generation. So because they're all out of 16, we get a 9 to 3 to 3 to 1 phenotypic ratio. Again, that's the phenotypic ratio, the characteristic, the look of the peas. Genotypic ratio would be different. So 9 to 3 to 3 to 1 phenotypic ratio. So we're wondering, Mendel's wondering, if genes are on different, Mendel didn't know about chromosomes yet, but we're wondering if genes are on different chromosomes, how do they assort into the gametes? Do they do it together or do they do it independently? And Mendel would have wondered himself, are these genes or characteristics, because he didn't know about genes yet, are they related? Are they linked somehow? Are they inherited together? So is it this, that the 2 dominant ones from one parent are always inherited together, and the 2 recessive ones from the other parent, are they always inherited together? Or is it this? Oh, does this look familiar? Finding the gamete allele possibilities, the combinations, using the FOIL method. So is it this, that you get more of a mix, yellow round, yellow wrinkled, green round, and green wrinkled? Which one would explain the data? So if it was this, remember we have to find our allele combinations, so we put them here, but in this case, we're stating that yellow and round are always inherited together, while green and wrinkled are inherited together. So if it's that, here's what our offspring would look like. And it would actually be a 3 to 1 phenotypic ratio. Is that what we got? No, we got 9 to 3 to 3 to 1. That does not work. So it must not be that. These genes or these characteristics must not be linked or related. They must be independent of each other, therefore inherited independently. So we're going to do a dihybrid cross the correct way. This is how the inheritance of these traits in peas actually goes. So this, again, is taking the F1s and then letting them cross-pollinate. Then we do finding the allele combinations using the FOIL method. Remember, first, outside, inside, last. So big Y, big R. Bring that down here. Big Y, little r here. Little y, big R for the inside. And for last, little y, little r. And then the second one, the second parent is the same because both parents have the same genotype. And then we start to fill in our boxes. So there's our first box. Keep filling them in. Let me know if you have any questions how these are being filled in. You're just taking all the letters, you put your Y's together, put your R's together. Big Y, big R, little r. Always write the capital one first when you put them into the boxes. So we'll fill those in. Now let's see how they look. The phenotypes. Lots of yellow round ones, but there's a yellow wrinkle. Oh, there's a green round, and there's a yellow wrinkled again, green round, and finally we get a green wrinkled. All right, now count them up. If you count the yellow round circles, there's 9 of them. Then there's 3 of the yellow wrinkled, 3 of the green round, and 1 of the green wrinkled. There it is. That's our 9:3 to 3:1 phenotypic ratio. That must be how inheritance works with those pea plants. At least those traits that Mendel chose to follow with 2 of them at one time. So the penguin says, bingo, we've got it. So from this, because those traits were not linked together and not always inherited together, Mendel came up with what's called the law of independent assortment, which we've already gone over when we went over meiosis. And this states that, remember how when chromosomes line up during metaphase 1, you have your homologous pairs coming together? Here they are in the picture, homologous pairs coming together. And what side do they go on? Are they on the left or the right? Can they flip-flop? Sure. So it's all random. You don't always have just dad's chromosome on one side and mom's all on the other. It's a random mix. So we can show that here. This one, we've got yellow and green. They didn't change with these two, but the goofy squiggly one changed from the left to the right, and either one's okay. It's random the way your homologous pairs, the different homologous pairs, the non-homologous chromosomes, how they align independently of one another, which means you get a good mix-up of the genes that are on all those chromosomes. This is Mendel's second law of heredity. It's called the law of independent assortment, and I am just completely amazed that he was able to come up with that, not knowing about genes or chromosomes yet in the 1800s. Again, that happens during metaphase 1. In this picture, we have all the blue on one side and the red on one side, but it could definitely have been flip-flopped. There is an exception to the rule. It seems like there's always exceptions to the rule that sometimes when genes are really close together on a chromosome, they will tend to be inherited together. They're so close that when crossing over happens, that they stay together in that chunk a lot and then end up crossing over together all the time. So it seems like those genes are linked because they're often inherited together more than usual. So a little small exception, like in this picture here, this chromosome has the dark pink and the light pink genes really close together. So often that chunk, if it does cross over and they do the place switch among the homologous pairs, then those two, the dark pink and light pink, will often be in the same chunk together and therefore be inherited together much more often than normal. We say those genes are linked. So two of Mendel's laws, the law of segregation, that you can't give both of your alleles to your kids. You can only give one for each trait. So we have meiosis to be able to pull those alleles apart from one another so that each gamete will just get one copy, because the diploid part of that will be reset when fertilization happens. The law of independent assortment, that is shown with the dihybrid crosses, and it shows that genes are independently inherited and usually not linked, with the exception when they're really close together on the same chromosome, that they may cross over together all the time and look like they're linked. Mendel was pretty lucky with his experiments, so he did have some good techniques, but you can also boil it down to some good luck, that he chose wisely with the individuals that he was testing on, and that was pea plants. He chose good traits that weren't linked, so those traits must be on different chromosomes and are inherited independently of one another. And he had strict control over which plants mated with who. Remember, you can control that with how plants will get fertilized. And took good data, good quantitative data, and did some good journaling as well, and then published his results. Plus, the traits that he chose were completely dominant over one another. So that's Mendel's complete dominance rules. There's going to be new rules coming up, so look out for that. The rules are going to change as we've gone into modern genetics now. We found exceptions to Mendel's rules. Let me know if you have any questions about all of that. But you will be going to do your dihybrid crosses now. So when you do your setup, if you have two that you're crossing and both of them are heterozygous for both traits, then you're going to have that full 4 different possibilities. You're going to need a 16 square Punnett square, and you'll use the whole thing. However, if they're homozygous for their trait, like what if the second one was big Y, big Y, big R, big R? Then you'd only have one possibility, big Y, big R, and you wouldn't need all 16 squares of this. So you'll get some practice in doing the problems that you're going to do. And again, let me know if you have any trouble. Refer back to your notes, and we will see you soon.